in this video, I'm going to talk about a type of cell division which is known as mitosis. So that's going to be the title of my video, mitosis. So as I mentioned before, this is one of the types of cell division, and one of the important facts to remember about mitosis is that it results in production of identical cells. So mitosis results in production of identical cells. So the cells, the original cell, also known as parent cell. So let's draw a quick picture. So parent cell. And imagine now it's got a couple of chromosomes. Obviously, in reality, in a human cell, it's not 2 chromosomes, but quite a few more. But this is just to represent kind of the idea of mitosis. So what's going to happen is you are going to produce copies of these chromosomes. So let me draw the next cell. So you're going to make these sort of copies. So 2 red sticks, 2 blue sticks, and then all you're going to do is you're going to move 1 copy of DNA into the resulting cell, and then the other copy into the second cell. And you can see that whatever DNA you started with, so I had 1 stick which was represented by blue pen, 1 by red, so that's exactly what I ended up with. So these resulting cells are known as daughter cells, and you can see that parent cell and both daughter cells are identical to each other. Now, as parent cell divides, what, as I said, essentially happens is you make a copy of DNA, and then you kind of split essentially that cell in half, and each of the resulting cells ends up with its own copy of all DNA the original cell had. As a result, the parent cell stopped existing, right? So it's no longer there because you split it in half. So this is something which people sometimes get a bit confused about. Then they think that at the end of the process you end up with 3 cells, but that's not the case. From 1 original cell you end up with 2 resulting cells. Now you can see that the actual essence of a process is not, I think, that difficult to understand, right? I mean, this is all summarized in this one diagram. I think the difficulty is the amount of terminology involved. As I mentioned in one of the previous videos, that's the kind of the tricky bit about learning about cell division. So even though the summary of the process is fairly straightforward, it's those details which make information more difficult to understand. But hopefully by the end of this video you'll feel a bit more confident about describing mitosis. So before we move on to mitosis itself, you also need to know about a stage in cell's life which is known as interphase. So let me write it down, interphase. And this is the stage in cell's life prior to cell division, in this case prior to mitosis. So this occurs prior to mitosis. And this is the stage in cell's life where it just lives its life, does its job. You know, if it's liver cell, it's maybe breaking down toxins. If it's skin cell, it's protecting you from viruses, from bacteria getting inside your body, etc. So it's just doing its job, living its life, and this stage is known as interphase. Now, as cell is gearing up towards cell division, what's going to happen in later interphase is that DNA is going to replicate, because you have to have 2 copies of DNA, remember, we saw it on that diagram, in order for our cell to ultimately split itself in half and end up with 2 cells. So during interphase, as I said, DNA, and also not just DNA but also organelles. Organelles replicate. And this happens just prior to cell division or mitosis. So once a cell goes into mitosis itself, mitosis is then kind of split, I suppose, and this is done by scientists, not like the cell kind of stops and thinks, so I better move on to this next stage. But scientists, through kind of observations of what happens during mitosis in the cell, they decided, well, let's just kind of artificially, if you like, split the whole process into stages. And each stage has got its own name. So then the whole of mitosis is, as I said, split into these so-called stages. So the first stage, which follows interphase, is known as prophase. Prophase. By the way, I am writing a lot of these terms with a capital letter just because I kind of treat it almost like a label or a title. But if it's in the middle of a sentence, it's all just lowercase letters, just to draw your attention to that. So prophase also, often in a lot of books, it's split into kind of two substages, which is early prophase and late prophase. But not necessarily. So you might come across some books where they just talk about prophase, but in some cases, as I said, it's also split into these two substages. After prophase, you have a next stage called metaphase, and then followed by anaphase, and then also followed by the last stage of mitosis, which is called telophase. And then finally, you also have another stage which is known as cytokinesis, or kinesis, I don't know, it just depends on how you want to pronounce it. Cytokinesis is the way I usually pronounce it. Now here, where it also sometimes gets a bit confusing, because in some books they say that cytokinesis is not part of mitosis at all, but it's a separate stage which follows mitosis. And in some cases, in some books, they actually combine telophase and cytokinesis into almost like one stage. And the reason for that is because in some books, they interpret mitosis as just division of DNA, of whatever is inside the nucleus, whereas cytokinesis is a division of cytoplasm, the rest of the cell, and that's why they're separating the two together out into kind of saying that cytokinesis follows mitosis rather than that it's part of it. Whereas in terms of chronology of things, actually cytokinesis and telophase often happens simultaneously at the same time. So that's why in some other sources of information, some books, they actually combine the two together. So that can be also just a little bit confusing, right? Just to add to, to the whole thing. Right, so what I'm going to do next is I'm going to draw some diagrams. You know me, I like drawing, just to explain things a bit more clearly. And we're going to kind of draw a diagram for each of these different stages. And I'm going to kind of tell you a little bit what happens during each stage. I'm not going to write a lot of information here at this point. So it's up to you to pause the video and kind of look at the diagram and maybe add some information, some descriptions as we're going through those stages. So the first thing we're going to do is draw a diagram of your typical interphase. Now you have to realize that the diagrams I'm drawing, they're very much simplified diagrams, because in reality, life is complicated. There's a lot of organelles in the cell, there's a lot of DNA in the cell. So I'm not going to draw every single thing. I'm going to sort of simplify and generalize things, just to highlight the bits I'm particularly, interested in at the time. Another thing which I wanted to highlight, we're going to keep using this red and blue just to represent 2 chromosomes, and just again to remind you that in reality you have 23 pairs of chromosomes, or 46 chromosomes, not just 2 or 4, right? So just keep that in mind. This is just representative pictures. So with interphase, you see the cell the way you would expect it to be, right? So it's just normal cell membrane, and then inside the cell membrane you've got cytoplasm and you've got your nucleus, and inside this nucleus during interphase you will have chromosomes which are a bit like this tangle, you remember, of DNA, so it's not really visible under the microscope, even though I've drawn them as the squiggly lines. In reality, it's going to be more like a grainy kind of picture. And so these bits of DNA are long and stringy. Maybe I'll draw them like even more kind of squiggly, folded onto each other. And these represent sort of molecules of DNA, right, before cell start to think, if you like, about division. So at some point during interphase, this DNA is going to replicate. I'm not going to attempt to draw a lot of squiggly lines, but what you have to remember is that each of those squiggles is going to have its own copy. So you have 2 copies of DNA. So each of those chromosomes are going to look more like that, right? So DNA then is replicated. So each squiggly line, if you like, is its own copy of DNA. So one chromosome is now made out of 2 copies of DNA, and the other chromosome is the same. And just again as a reminder, they're connected to each other by this little kind of dot, if you like, a little connection called centromere. Let me write it here because I haven't got quite enough space. So it's centromere. So that's really what happens in DNA, in interphase. And also a reminder that also organelles are going to double up, they're going to replicate, but again, I'm not going to draw that because it'll make drawing really complicated. So then the next part, the next stage is going to be prophase, right? Just to remind you. And for my drawings, I'm actually going to separate early and late prophase out. So let's start with early. So in early prophase, the key thing which happens is that you remember that instead of these long stringy bits of DNA, what I need to do is make them more compact, because if I don't have them in a more compact shape, I won't be able to easily move those chromosomes around the cell. So that is the next stage which needs to happen. So again, I'm going to draw my cell. Maybe I can try to draw it a bit bigger. Okay, so you can more easily see what's going on. And again, I'm drawing nucleus super big, just to give myself a bit more space inside my drawing. And so what's going to happen is, in early prophase, instead of these chromosomes being long and stringy, they're going to get compacted, and this process is known as condensation. So what you're going to have is those chromosomes are going to turn into these more compact shapes. So they're no longer looking like that, but rather like that. And that's the point where your chromosomes become visible under a microscope, so you can actually see those chromosomes. So the key sort of event here is chromosome condensation. Again, notice I'm not writing very much, I'm just giving you some key words, so it's your job to explain this process in more detail and expand on it and maybe do a bit of independent research into it. So that's one of the events which happens. Now, another important event which takes place in early prophase is that you start to have this structure being formed called spindle. And spindle is going to be a collection of like little fibers which are going to help us to move chromosomes around the cell. So the spindle is going to start forming at one end of the cell, and it's going to look a little bit like that. So a lot of kind of parallel lines all gathered up between these two kind of structures called centrioles. So you've got two lots of centrioles, and as they are moving apart from each other, they are producing this like a web-like sort of parallel tubules, which are collectively known as spindle. So in early prophase, the spindle is only starting to form, and as I draw the next picture for late prophase, you will see how that's going to progress. So my next picture is going to be for late prophase. So in this stage, I'm going to try to fit it in here so we can see sort of direct comparison between early and late prophase. So again, I'm going to draw my cell membrane, try to keep everything as big as possible. So what I'm also going to start drawing here is I'm going to show that the spindle now is grown. So the centrioles are still there. Centrioles are just going to sort of simplify them as two like kind of ovally shapes, and those fibers are now extending all the way across the cell, a bit like that. And my chromosomes are now going to be connected to those fibers which make up spindle, and they're connected through the centromere. So that middle bit, you remember centromere, that dot, if you like, that's what's going to be connecting chromosome to the spindle fibers. And another event which I hope you can pick up from my drawing is as spindle is growing, then nuclear membrane, which is that black line which was surrounding chromosomes in interphase and early prophase, it is disappearing. So that nuclear membrane is going to start disappearing, and maybe I'll do it like a little kind of dotted line. It's going to start disappearing in early prophase, and it's going to finish disappearing in late prophase. So by the end of late prophase, it's completely gone. So another thing which I wanted to mention is that I'm obviously drawing these stages as little kind of snapshots in time. And the reality is this is a dynamic process, so it's a bit like an animation where things are merging, where things are slowly disappearing or slowly moving, and one stage is kind of very gradually becomes the next stage. So there's often, not exact moment in time when early prophase becomes late prophase, or when late prophase becomes metaphase, etc. So it's all kind of a slow process where things kind of, as I said, merge from one stage to the next gradually. Okay, so from late prophase, we're moving on to the next picture. So I'll move on to the next sheet. I'm going to keep this one up here. So the next stage is metaphase. So again, I'm going to draw the cell. I'm going to draw my two lots of centrioles. The spindle fibers are still there. Also, each spindle fiber is often known as microtubule, but collectively all these microtubules are known as spindle. And during metaphase, what's going to be happening, or rather, this process is going to start actually in late prophase, the spindle fibers are going to pull and push those chromosomes towards the midpoint in the cell. So the chromosomes can't move, if you like, by themselves. So instead, the spindle fibers work a little bit like strings on a puppet, I suppose, and centrioles like a puppeteer, which is just pulling on those strings by elongating them or getting them shorter, and moving chromosomes in a specific position within the cell, wherever they need to be, depending on what stage, which stage of mitosis it is. So imagine now that this red chromosome is going to start getting moved towards that midpoint, and so in metaphase it's going to end up in exactly kind of that halfway point within the cell, and the same is going to be happening to the blue chromosome. So this one is going to get moved towards the same sort of midpoint position, so it's going to end up around about that there. So that midpoint position is just an imaginary line, right? And it's often known, that line, as a metaphase equator or metaphase plate. So metaphase plate or equator. By the way, the ends of a cell, right, so where the centrioles are often known as poles. So this is one pole, this is a different pole. So it's a bit like sort of think about a cell as if it was a planet Earth, I suppose, right? So the midpoint is equator and the far ends are poles. Right, so that's your metaphase. So you're now aligned all your chromosomes along this imaginary midline or equator. And what you then going to start doing is you're going to start moving the two copies of DNA. You remember each side of our chromosome has its own copy of DNA, so we need to separate them out so that each, you remember each copy is going to end up in its own cell. So you've got to separate those copies into two lots. And that's what's going to start happening in anaphase. Anaphase. So next picture. So cell again. I'm still going to draw centrioles. Still going to draw spindle. But I'm going to now show that my chromosomes are going to start separating. So that red chromosome, the two sides or two sister chromatids, just reminds you about some of the terminology, right? So those sister chromatids are going to get separated and they're going to start traveling to the opposite poles of a cell, right? So one half of a chromosome is going to start moving this way, the other half, the other sister chromatids is going to start moving towards the opposite pole. And the same story is going to be happening with the other chromosome, right? So you don't need to draw the arrows pointing which way it's moving, but just for kind of, to make sure that we're all on the same page. So that side is moving towards this pole, and the other two, what used to be sister chromatids, are now moving towards the other pole. Now, once while they were attached to each other, those chromosomes, the sides of the chromosomes, were known as sister chromatids. Let me just remind you again, so maybe I'll draw a separate quick picture. So that was our chromosome. And these two sides were known as sister chromatids. But once they're separate, we can't call them sister chromatids anymore because they don't belong to the same chromosome. So they're then known as daughter chromosomes. So these are daughter chromosomes. So once again, what used to be sister chromatids, once separate, they become known as daughter chromosomes. And so they keep traveling, in opposite directions until they reach the poles of that cell. And this stage is then known as telophase. So I'm going to draw a couple of pictures actually for telophase. So let's do this one first. So you still have your spindle fibers, but this time that daughter chromosome got all the way to the end of this pole, the other daughter chromosome got to the end of the other pole, and the same happened to the blue chromosome. So once we reach the end of the poles, that's now considered to be telophase rather than anaphase. So you can see it again. So red chromosome, one daughter chromosome here, another one there, and the same story applies to the other blue chromosome. So once that is done and reached, then you still carry on with telophase, but then the pictures start to change, because what you're going to start having is the spindle is going to start breaking down, so it's no longer there, and those chromosomes are going to then straighten out like that. And another event which is going to start happening is that the new nuclear membranes are going to start appearing around these chromosomes at the two ends of the resulting cell. The cell is still together, so it's still single cell, but it's starting to have these two separate nuclear membranes. So at that point, you are finally reaching the last event of the cell division, which is cytokinesis. And at this point, the nuclear membranes are going to be fully formed. So let me just draw those first. So chromosomes are now inside those nuclear membranes. But what's going to start happening also is that cell is going to start slowly splitting in half. So the cytoplasm also need to separate. And the way it separates is that you're going to start having this kind of pinched effect, I suppose, right? Where that midpoint is going to start getting thinner and thinner and thinner until the two sides come together and you end up with two completely separate cells. And this kind of pinching, if you like, action, that's a part where the cell membrane is starting to get a bit closer to the other side. This is known as division furrow. Also in some of the older books it's called cleavage furrow. Right, I hope you can see why, and I wouldn't be surprised if it was a male scientist, right, who came up with this name. So at the end of the whole process then, at the end of cytokinesis and at the end of the whole of mitosis, what you're essentially going to end up with are two separate cells, and those chromosomes, they're going to again start decondense. I'm going to go back to this kind of squiggly, sort of long molecules of DNA. And what you're going to end up with is two identical cells. And so that sort of summarizes the process of mitosis for you from start to finish.